Okay, so I'm going to go ahead and get started. I'm probably not going to go for the whole time we're supposed to have class today. I just want to go through as much as we can. I don't want to make this too incredibly boring for everybody. I know this is not the easiest way to learn. Hold on, let me mute everybody. Hold on. Okay, let's mute. Okay. I know this is not the easiest way to learn everything. I know this is not optimal. We're going to try our best to get through everything. I do want to make a couple of announcements about class. At the end, if you have some questions about any of the assignments, right now, just assume that I'm going to be super flexible with any of the due dates right now. I'm meeting with Raina today to go over some stuff for lab. I'll send you guys an email. I have been giving permission to let you guys come in as individual groups to come pick up supplies, work on your independent projects. We just can't all be in the lab at the same time. So I'm going to send out kind of a scheduler thing on SharePoint to have set times where you guys can come in. If for whatever reason you're not available to come in or you don't want to come in, that's okay. Just let me know. We'll try to work something else out. But we're going to be really flexible with the due dates. I still would like to get a poster. We're obviously not having research day, so that's not going to happen. But I would still like everybody to have a poster, have that opportunity to do a poster, and then hopefully we'll do our unknown projects. We'll see how that goes. Most of the unknowns is really just inoculating tubes and then looking at how they change. So I may do some inoculations for people, post some pictures of how the tubes look like. I'm still planning on doing a lab practical as long as we can come back. That I will let you know if I do cancel that and we have to do some sort of alternatives. Right now, the exam 3 was scheduled, I believe for the 3rd of April. Just leave, don't worry about that date. We may do it if we get to come back on the 13th, we may do it that 14th. But I will let you guys know if we have to do some alternative online kind of stuff. For today's lecture, I'm just going to go through everything. There won't be any additional assignments this week. It'll just be those lectures. At the beginning of each week, as things progress, I might have a couple of assignments for you guys to do to make up some of the lab time, depending on what lab assignments we are allowed, we can and cannot do. So just look out for emails, look out for posts on Moodle. I'm going to try to update the schedule this week and kind of get everything set up. Since we're going to do viruses early, we'll do the other ones in the later weeks on there. And we're all just trying to figure this out. So please ask me if you guys have any questions about anything. I'm trying to be as flexible as I can with any of the due dates of anything that needs to be posted. Right now, nothing is due. The next assignment to be due was the poster. So that's the only thing you needed to worry about, but don't worry about that at this point. Okay, so let's talk about viruses. And I feel very weird talking to myself in my office, so this is kind of awkward. Okay, so let's see. I'm going to try to draw on the screen a little bit too. I know it's not the easiest thing to do to kind of highlight some things that pops up. There is a version of this PowerPoint on Moodle posted on there, so you guys can download that when you get a chance. So let me switch on there. Okay, move that out of the way. All your wonderful faces are right here. I'm just going to move them. Okay. All right, so fun facts about viruses. So the reason I moved the virus stuff up here is that clearly everything and anything that we're doing right now is revolving around this virus. So it's kind of big in our lives right now. So I thought it would be good to give you guys a little bit of information. I mean, we were going to talk about this anyway, but it is nice to have it at least available for you if you're questioning anything. Okay, so fun facts about viruses. So in the 1800s is when we first were developing and finding viruses. They called it filtrate. So at the time they knew about bacteria, but they didn't know about these kind of infectious fluids is what they called them. They passed them through a filter that removed all bacteria, fungal things, but that filtrate was still able to cause disease. And then in the 1890s, is when our first virus was characterized, and that was the tobacco mosaic virus. It causes a disease of tobacco plants, and it causes portion of the leaves to start dying. So they call it a mosaic virus because the leaf looks like a pattern of dead and alive tissues. Now, for those of you who don't know anything about cigarettes, tobacco is grown in these big giant leaves, and it's used to make cigarettes. And so if half the leaf was dead, it was kind of a big problem for the farmers, and it was not an optimal thing. So they were very concerned about this. So they first discovered that virus was causing that disease in tobacco plants. Now we know there's viruses out there that affect every sort of organism: plants, animals, algae, protozoa, fungal organisms, even bacteria get infected with viruses. Viruses cannot live, when they're not technically alive, they cannot live or replicate outside of a host. So we call them obligate intercellular parasites. Now, you guys have probably heard a lot about the coronavirus. You probably heard a lot about flu and even common cold, about living on surfaces. Now, virus particles can get on a surface, they can get on different surfaces, and they can survive to infect for an extended period of time. Now, for every virus, that's a little bit different. We don't really know about this new virus exactly how long it can live on the surfaces. We're thinking it's somewhere between 24 and 48 hours. The first tests that have come up has said that. So we're still testing and figuring that out. How long can it live on surfaces? And usually when they do these tests, they'll do plastic and steel and fabric and all those things. Talking too much, I'm already coughing. I promise I don't have coronavirus. I took my temperature this morning. I'm normal. Okay. So they're obligate intracellular parasites, but they can survive on surfaces. Every virus is a little bit different depending on how long it can live on the surface. Viruses are not what we call alive, though. They're not technically alive. They can't replicate on their own. They have to use the host machinery in order to replicate and move on either to another cell in the host or to another host. Okay, let me pull that out of there. Okay, so here's just some general comparison between viruses. Get that there, okay, easier to see there. Between viruses and a typical bacteria. So not all viruses will have membranes. We'll talk about the structure of viruses in a little bit. Some of them do have membranes, but it's membranes that they have stolen from eukaryotic cells. They do not produce their own membranes. Their genomes can be either DNA or RNA, but they use host machinery in order to replicate their genomes. So they don't necessarily have any of the like DNA polymerases or RNA polymerases. Some viruses we'll talk about will have a special type of enzyme called an reverse transcriptase, but most of them don't have their own proteins for replication. So they will hijack the host proteins in order to do that. They are not sensitive to antibiotics. So if you took an antibiotic, you would not kill a virus. Antibiotics are specifically for killing bacteria, and they affect bacterial cells. They do not affect viruses. As far as the size of viruses, viruses are what we consider ultra microscopic, meaning that if we used our light microscopes upstairs, we would not be able to see a virus underneath those microscopes. Although my son has tried to ask me a couple of times, can he see the coronavirus if we found it? And I kept telling him, no, we can't. But there are special electron microscopes that can visualize viruses. That's all those pretty pictures you've been seeing out there. Some of them are artist renditions of the coronavirus. Some of them are actual electron microscopes of viruses. So the smallest viruses that we know about are the ones that infect bacteria. Those are bacteriophages. We talked a little bit about those before. So those are bacteriophages. A couple of the viruses on this list. Get my pointer here. Although I'm seeing staying on the screen, so maybe I should. Can I erase? Let me erase that. There we go. Erase that one. Okay. What does that do? Oh, that's moving things around. I don't want to do that. Okay. Draw. Let's do that. Line. Okay. let's try this. All right, so these are the different types of viruses. A rhinovirus, that's one of the viruses that causes the common cold. We'll talk a little bit about the names. There's that tobacco mosaic virus that I talked about. And then so they go over, and then this is supposed to represent over here on this side, that's supposed to be an E. coli. And then a red blood cell you can see. And then you guys' face is on top of mine, so I can't see it over there. Ebola virus is a little bit one of the bigger of the viruses. This is the smallest known bacteria over here, chlamydia. So that's just a comparison of all the sizes. Now, where I saw those things? So, mouse click. This is just a similar picture. It's just showing a couple of different viruses that were not on the other picture. Herpes simplex, rabies, HIV, streptococcus, one of the bacteria you guys would have looked at underneath the microscope. You can see the comparative size. One of the bigger ones is the pox virus, but we are still not able to visualize it underneath our light microscopes. All right, so let's talk a little bit about a viral life cycle. How does the virus replicate? How do we get a virus? How does it cause damage to the cells? So just in general, now, depending on the type of virus, it will maybe have a couple of different things, different stages than this kind of simplistic drawing. This kind of gives the general stages of how a virus attaches and replicates. But we'll talk about a couple of additional steps for some viruses that are a little bit varied from this life cycle. So somehow the virus here, this little virus here is supposed to represent a little bit of DNA covered by protein, needs to attach to your cell. Now, depending on the type of virus, whatever type of cell it attached to can be different. We'll talk a little bit about attachment when we go specifics into the life cycle. But depending on what type of virus it is, it will attach to different cells. So a virus could attach to cells in your lung, can attach to cells in your skin, and your immune system, like HIV. So it just kind of depends on the virus, what type of cell it attaches to. But in order for it to go in, it needs to attach to the cell. So the virus normally uses a receptor on the outside of our cell that has a normal function. And then once it attaches to that receptor, the virus enters the cell. There's a couple of different methods for entry. We'll talk about what those are in a little bit. Then within the cell, the genome of the virus, whether that's RNA or DNA, will be released. The virus will use the host machinery to express those. So depending on whether it's a DNA, an RNA, it'll either go through the process of transcription, translation, or just translation. And then that host will put the viral pieces together. And the virus will assemble. So this is viral assembly here, and then it will release. Depending on the type of virus, it might kill the cell. It may not kill the cell. It may replicate and just release into the cell or into the bloodstream or wherever this virus seems to be attaching to, and will travel to another cell and infect it. Let me erase all those. Okay. Okay, next up, go. All right. So the viral components. So what are the components of a virus? Well, the most basic of the viruses are going to have two components. They're going to have their genomic material, which is either DNA or RNA, and then they're going to have something surrounding that genetic material, either just protein or protein with an envelope on there. So you can see on this slide it says not envelope is not found in all viruses. So some viruses do not have an extra envelope, but all viruses will have an RNA or DNA genome and then some sort of protein attached with that genome. So let's look at those viral components. So they're not cells. Viruses are not cells and they don't look like cells. So they won't have things like an endoplasmic reticulum or a nucleus or a Golgi or anything like that. They will really just be protein and nucleic acid. So they'll lack all those cellular components. So no ribosomes, no DNA polymerase, really very, very basic. And they're very simple structure, usually the capsid, and the capsid again is made out of proteins, and then an envelope for some of the viruses. It will have the nucleic acid molecule, either DNA or RNA, and some additional proteins depending on what type of virus it is. So if we see these little pictures right here, this one is called, get my little drawing tool, the naked nucleocapsid virus. So that, when we call them naked viruses, that means they don't have an envelope. They're just capsid, which is protein, and nucleic acid, DNA or RNA. Where if we have an envelope virus, which is this guy over here, then we've got this lovely envelope on the outside here, like our friend the coronavirus and HIV. And that envelope is stolen from the host cell. So they don't have the machinery to make their own envelope. They steal it normally on release from the host cell. And these are just some pictures of different types of viruses and how we classify them. We're going to go into those different types of classification. The first one being icosahedron, enveloped, and then helical. We'll talk about each one of those. Oh, let me delete my, clear all drawings. There we go. Okay, got that one down. Okay, all right, so let's talk about these different capsids. Okay. So the general structure of the capsid are repeating protein subunits called capsomers, and the capsomers are self-assembled into that capsid structure. And we'll classify these different viruses based on the capsid structure. So the first type is a helical capsid. So the capsomers are these little, they almost look like little pills here. The little pills, and they're stuck on top of each other. And they're forming this tube structure here. And then here's the genetic information, the nucleic acid. And the nucleic acid is being wound in the middle of the capsomers as they form into this capsid. So the capsomers are like the little individual bricks, and when they all form together into the helical structure, then that's called the capsid. Some examples of helical capsid viruses are tobacco mosaic virus, the one we talked about. And you can see the long tubes right there. Those are long tubes, and if we zoomed in on them, they would be just the capsomers stacked on top of each other with the nucleic acid information right there in the middle. And then they're stacked in these long tubes like that. Influenza is also a helical capsid, but it is not a naked one. It's enveloped. So on the outside, so inside here, you can kind of see that little red section right there. That is supposed to represent the helical capsid, and then with the genomic information in the middle. And then on the outside is the envelope. I'm going to talk a little about influenza on Thursday, I think. Probably not going to have enough time today. I don't want to have you guys here for like all the time, so I wanted to save that probably for Thursday. So we'll talk about influenza too, because influenza is feeling kind of left out right now. Usually they're it's very popular right now, but nobody's talking about influenza. Okay, so those were the helical capsid. This is the icosahedron capsid. So you can kind of see that structure is more kind of like this crazy pyramid, kind of a cool looking diamond shape almost. So well, it's called an icosahedron, haha. So they're 20-sided figures, and they have 12 corners in them. So they all have that kind of same cool icosahedron structure. The capsomers are a little bit different on those corners as opposed to the sides here. So the sides are all stacked up like that, and then they have the little corner proteins. Adenovirus is an example of one of these. And the number and arrangement will be a little bit different depending on the type of virus it is. So the capsomers might be a little bit bigger, smaller, so there'll be less of them or more of them depending on the type of virus. So these guys are the icosahedrons. lipids. So that's the capsid. Again, capsid is made out of proteins. Then the viral envelope is made out of phospholipids. It is just like our phospholipid bilayer because it is stolen from our phospholipid bilayer or from another eukaryotic cell, depending on where the virus is coming from. So as the virus releases, so part of that releasing, it can take part of the host membrane with it. So some will bud off the cells, and then others will leave through the normal, ooh, that was a bad drawing, okay. The normal route versus the ER. So as they're going through the endoplasmic reticulum, they're getting their own membrane as they're coming out of the cell. In the envelope, on the outside, and you probably noticed this if you've seen pictures of the coronavirus or an influenza, on the outside of the envelope are proteins, and normally they're glycoproteins. So they have a little bit of sugar and protein that stick out from the cell. And I'm gonna draw a really bad drawing of that right here. So we got the envelope, and then we've got these little guys that kind of, oh, that's really bad. All the coronavirus pictures. Anyway, those are the little spikes that stick out from the cell. And depending on the type of virus, they'll probably use these spikes in attachment. Okay, so this will be what they've used to attach to our receptors. So HIV, for example, influenza, coronavirus, I'll use those spikes to attach to the host cells. So when it's forming the envelope or stealing the envelope, these proteins will get stuck on the outside of that envelope so that they can use when they attach to their next cell. So they're assembling, they're going to release somehow, and then they're going to go move on to their next cell, whether that's in your body or in the next person's body. This is an example of herpes simplex virus. As I'm going through, I'm mentioning a couple of different viruses. If you guys are interested in doing any of these viruses for your independent microbe, we're still planning on doing that project. Just let me know. I'm going to set up a table so you guys can populate with whatever bacteria, virus, fungal infection that you guys want to put on there. If you're like, yeah, herpes simplex, that sounds fantastic, I want to learn more about that, you can pick that as your virus. We just don't want to have any duplicates, so the sooner you put something in there, you can hold your spot. Okay, so this is herpes simplex. It has the icosahedron capsid. So that's right down here. It's got the envelope, and then it's got those little glycoprotein spikes. The genome on the inside of the capsid there, right? So this is an artist drawing of it. This is what it looks like on an electron micrograph. So the capsid on the inside here, and then the envelope here on the outside. So here herpes simplex virus. There's a lot of different versions of herpes simplex virus. It's one of the viruses that once it affects can go latent, what means it just kind of hangs out for a little while. We'll talk about latency at the end. But it's a very interesting virus. There's a couple of different types of them if you are interested in doing herpes. Okay. Complex viruses, they are ones that have a little bit different than just the envelope or the capsid. And this includes, these are a couple of examples right here, pox virus that have these lipoproteins in them as well. So they have a little bit different structure. This is not typical of viruses. This is a very unique virus. This would be smallpox, cowpox, pox viruses. And then down on the bottom, which we've seen before, is the bacterial phages. And they have that crazy kind of spider-like structure, which is very, very unique for viruses. So it's got the icosahedron head here with the viral genome in it, and it's got these crazy tail fibers that are used to infect bacteria. So these guys are a little bit different than everybody else. They have kind of a different structure, a little bit more complex than just protein, nucleic acid, and maybe an envelope. Okay, so how do we classify bacteria? You'll, as we're going through the different types of bacteria, viruses. As we go through the different types of viruses, you'll see these names come up. So for example, the genus name, it will have some sort of virus at the end of it, and the family name like Coronaviridae, we'll talk about. Others will have ALES at the end of it. So depending on the type of virus, you'll see that kind of nomenclature of the taxonomy of them. So viral species, if we call them a species, that means they share similar genetic information. They all have RNA as their genome, and they all infect the same host. Okay, we'll talk about how viruses kind of evolve, because they do, and they do very quickly. And then a subspecies will be designated by a number. When we talk about coronavirus, we'll talk about that, about how they've been given a new number within that species. All right, so the viral genomes, there is a couple of different options of viral genomes. They can be made up of DNA or RNA, but not both. So they can have a double-stranded DNA genome, a single-stranded DNA genome, a double-stranded RNA genome, and a single-stranded RNA genome. The number of genes on the genome of the virus varies depending on the virus. So some have a little bit longer genome with more genes in it than other viruses. Depending on what type of replication it does, what type of host it infects, it'll vary a little bit, but it's not a lot. It's not like the size of the E. coli genome. It will be very few genes. Just for the basics. How do I invade? How do I hijack the host machinery? How do I make new viruses? And how do I release? So that's all the virus cares about. Here are some of the different types of viruses and if they're RNA or DNA. So Ebola, our lovely friend in there, influenza, HIV, norovirus. If any of you have had norovirus, it is probably the worst food poisoning that you have ever had in your entire life. It feels horrible and it's super contagious. Poliovirus, which we thankfully have eradicated in this country. DNA viruses include herpes virus, we talked about adenovirus, papillomavirus, that's HPV, human papillomavirus, parvovirus, infecting other organisms besides us. Okay, so those are kind of general different types of viruses in those categories. This is just another example of a couple of those different types of viruses and what kind of genomes they have. So we've got the double-stranded, again, RNA, single-stranded RNA, double-stranded DNA, single-stranded DNA. Okay. So a couple of highlights on here. Here's our coronaviruses right here. And as you notice, it causes mostly the common cold, right? And then our SARS there. They are single-stranded, enveloped RNA viruses. Rabies is up here, measles, influenza, HIV, retroviruses. Rhinoviruses also cause a common cold. If anybody's interested in doing the common cold, that would be one of the viruses on there. Any other ones exciting in there? Herpes, again, these are down on the DNA, herpes virus, smallpox, adenovirus. Okay. All right, so let's go a little bit more in detail on the different replication stages of the viruses. So just to remind yourself again, so attachment. So the virus is going to attach to the host cell. It's going to get inside the host cell. It's going to uncode its genome. It's going to replicate its genome. It's going to make all the proteins needed to make a new virus. And then somehow it's going to release. Okay. This little table is from another textbook. It's just talking about the difference between bacteriophages, so infecting bacteria, and animal viruses. This is just, again, another summary. I'm going to go through each one of those little parts and give you some examples, maybe of some of those steps in there. So let's talk about attachment first. Get my drawing here. Okay, so invasion of the virus. Let's think in our minds here right now. We'll use the common cold as kind of our example. Someone sneezes near you, like my 2-year-old likes to do quite often. You get some virus particles they enter your body through one of the various portals of entry, through your nose, through your mouth, can even get it in your eye, depending. So that's viral invasion. Now, once the virus gets in, it needs to find its way into the location where the cells that it can attach to are, okay? So you can imagine if it's a common cold, we'll talk about like a rhinovirus. The cells that a rhinovirus attached to can be in your nasal cavity. So if you inhale those, they can infect those cells inside your nasal cavity. It's fairly easy for them to infect. Whereas another virus may have to travel through your gastrointestinal tract, make it down to your intestine before it can infect those cells. So it has to get in, it has to invade, and then it needs to attach to the correct host cell. So the virus particles will attach to the receptor, and the virus particle attaches to that receptor, and it needs to have the correct receptor on that cell for the virus to attach. If there's not the correct receptor, the virus is unable to get into that cell. So the rhinovirus has the receptor, the correct receptor is on the cells in your nasal cavity, but if it got on your skin, it's not going to infect your skin. It doesn't have the right attachments on those skin cells, so it's not going to get inside your skin. So the cold virus, if it lands on your elbow or your hand, it's not going to infect you until you touch your face. We're not supposed to do that anymore. Until you actually enter it into your body. And this is what the idea of not touching your face, like all these things, wash your hands, is that your skin is a really great barrier, and most of those types of viruses are not going to infect anything on your skin. Now, there are viruses that can, but respiratory viruses infect your respiratory system. So bringing them from your hands to your face, that's when you're going to infect your cells, bringing them into the route where they can get into your body. So most of the time, people don't know how much you touch your face. And so you want to wash your hands as much as you can so that when you do touch your face, you don't potentially have some sort of virus on your hands. Okay, so virus gets in, finds the correct cell, and the receptor on the cell, on the host cell, hooks up with maybe one of the spikes on that virus, and then the virus is able to enter in the cell. So viruses will only infect host cells which they can recognize and bind to those receptors. That's very important. So only certain viruses can only infect certain hosts. Now we'll talk about how this can change, but normally a human virus is not going to be able to infect a dog, a cat, normally, but this can change. So normally it's only going to infect one type of host and stick with that host, because our cells look very different than cat cells or dog cells. So they might not have the correct receptor so that virus can get in. So we call that the host range. The host range normally is one species. Sometimes with things like influenza and coronaviruses, it can be more than one species. But most of the time, human viruses are going to infect humans and not other species. So we call that the host range. Then they only infect specific tissues, so like cells in your nasal cavity, lung cells, skin cells, intestinal cells. And we call that tropism. Tissue, what's the specific tissue? Sometimes that can change as well. Okay, so we look at host range and tropism based on what type of cells the virus can infect. So let's look at an example here. This is HIV. So some of you probably have looked at HIV a little bit. We'll get into HIV a little bit more. If you want to pick HIV as your microbe, you're welcome to do that as well. My goal is once everybody's picked theirs, I'm going to put together a list of things people haven't picked so we could talk about them in class. Okay, so HIV. HIV is an RNA virus. It's considered a retrovirus. It reverse transcripts its RNA into DNA. That's why it's called a retrovirus. And it has the spikes on the outside, one of them being GP120 right here. And in order for HIV to infect cells, it attaches to a couple of receptors that are found on a very specific type of immune cell called T4 or T helper cells. So they have this receptor called CD4, and then HIV can use 2 additional receptors, CCR5 or CXCR4, which are also found on the T cells. So HIV can only infect cells that have the CD4 receptor and one of these 2 co-receptors. If it interacts with another type of cell that doesn't have those receptors, it's not going to infect that cell. Okay? Now, mostly these receptors are only found in T cells. So CD4 is specific to T cells, therefore HIV only infects T cells. So the tissue tropism for HIV is T cells. For those of you guys that are also taking immunology, we're going to talk a little bit about sometimes they do infect macrophages, because sometimes macrophages also have CD4 receptors on them. But kind of as general, HIV just infects T cells because of that tissue tropism idea. I'm going to talk about this again when we were talking about coronavirus at the end, but viruses can change fairly quickly, especially those that have RNA as their genome. They can withstand mutations very, very well, and it's actually beneficial to the virus to have a very high mutation rate. Because if they mutate those glycoproteins on the outside, it's potential that they can infect a new type of cell. So the host range and the tropism can change if the virus mutates the outside of it so that it can infect a new type of cell with a new different type of receptor. And this happens quite frequently, as it did happen with the coronavirus, or at least we think it did. Okay, so attachment. Okay. It attached. Now the virus needs to gain entry into the cell, and it needs to uncoat its genome so that the genome can be exposed to all the host machinery. There's 2 ways that the virus normally gets into the cell, usually through the process of fusion or endocytosis. So let's look at those. This is the picture from your book. I'm going to give you guys a couple of different pictures as well that are a little bit like actual pictures, like electron microscopes. Okay, so here's the binding to the host cell. We've got the interaction of the viral spike here with a receptor on the cell. Then we have fusion with the host membrane. So you can see that really bad at this drawing thing. Okay, the envelope of the virus is fused with the host cell, and now the virus can release its capsid plus its genome into the cell. Okay. So that represents fusion with the host cell membrane, and then down here is endocytosis, where the virus has tricked the cell into bringing it in through that process of endocytosis. So virus comes in to the cell, then releases its capsid and its genome. So here are a couple of electron micrographs of that. Let me clear all those drawings out there. So this is fusion of the viral envelope with the plasma membrane here, and then here is the receptor-mediated endocytosis on this side. That's bringing that virus particle, so the yellow-blue guys, the virus, bringing it into the cell. This just gives you a little bit different picture of that same thing that your book shows, a little bit more stepwise. So here is the attachment of the virus to the host cell membrane. That's supposed to be the interaction of that spike with the receptor. This is only going to happen in viruses that have an envelope. Okay. Viruses that just have a naked capsid can't do this whole fusing because it doesn't have the envelope to fuse with the host cell envelope. Okay. So those two attach. They are stuck there for good. The membranes begin to fuse. You can see the membrane of the virus is kind of flowing right into the membrane of the host cell, allowing it to come into the cell, and then eventually releasing its genetic information. Endocytosis, again, tricking the host cell into bringing it in. It attaches to the host cell. It's bringing it in there. And this can occur with both enveloped viruses, which is up here on the top, or naked viruses. So this one does not have an envelope. It's attaching to the receptor. The receptor is binding to that glycoprotein, or the proteins on the outside of this virus, then it's bringing it into the cell through endocytosis. So again, this could be naked or just envelope viruses can do that type of entry. Okay, now biosynthesis. So the virus is attached, it's brought into the cell, and now it's going to start the process of making new viruses. So biosynthesis includes replication of the genetic information and then creating any proteins that are needed to have that process go on. So for example, when talking about HIV, that reverse transcriptase changes the RNA genome into DNA. So the free nucleic acids, so the DNA or RNA, can take control of the host machinery, our DNA polymerase, our RNA polymerase, our ribosomes, etc., and use it to make new viruses. The process of doing that is a little bit different if the genome is an RNA, and there's two types of RNA we'll talk about in the next slide. RNA we consider positive, and RNA we consider negative, and whether it is a DNA virus. Okay, so the process is a little bit different depending on the viral genome. I like this picture. Ooh, let me clear all the drawings. From your textbook that gives a good summary of all the different types of viruses and how the replication process happens. So let's walk through that. Okay, so this is a single-stranded RNA, so one strand of RNA. Positive strand RNA, and then here the negative strand RNA. Positive strand means that the ribosome can read it and make it into a protein. So that's why it means by positive strand. If it's negative strand, it's the opposite, and the ribosome can't read it. So our negative strand RNA has to be changed into positive strand RNA before this little pretzel-like thing is supposed to represent a protein. So the only type of RNA that can be translated by the ribosome has to be positive. So if we're just a positive strand virus, it's ready to be read by the ribosome. It doesn't, any additional steps don't need to happen, and it can be made into the protein, okay? If it's a negative strand RNA, then it needs to be converted somehow into a positive strand mRNA so it can go into the process. Positive strand RNA can also be converted into DNA and then incorporated into the host DNA and just replicated like the DNA of the host. Okay, so it just gets embedded in the host processes here. So this little box here, I think, is trying to represent what the host is going to do to the genetic information. So single-stranded DNA can also be incorporated into the genome. Double-stranded RNA viruses, the positive strand of that double, because so the light green one is supposed to represent the negative strand. So a double-stranded RNA is going to have a positive negative strand. The positive strand can then be used just into the translation process. So depending on what type of genome you have as a virus, we have to go through some extra steps before we get to the viral proteins that we need to make a new virus. Okay. With DNA viruses, they replicate their DNA using the host machinery. So our DNA polymerase uses to replicate the DNA. And then the synthesis of capsid proteins is used by the host machinery. That DNA gets transcribed into RNA, like the host RNA gets translated into protein, just like host protein. With RNA viruses, that's when we need an additional step, possibly before it gets transcribed into the appropriate protein. Some host cytoplasms, the virus will bring in what we call an RNA-dependent RNA polymerase. So our RNA polymerase, the one we've talked about, the RNA polymerase that our host cells use and bacterial host cells use, we call a DNA-dependent RNA polymerase because it uses DNA as its template and it makes an RNA. An RNA-dependent RNA polymerase uses RNA as a template. So it's a different, it's kind of outside the central dogma that we're used to. Okay. So again, as we looked at that last picture, depending if the virus has a positive strand RNA or an anti-sense negative strand RNA, it will use maybe one of those host derived proteins in order to change into the appropriate genome. Retroviruses are a very specific class of RNA viruses because they use a protein, a specific enzyme called reverse transcriptase. And reverse transcriptase, oh, this is going to be really ugly, uses RNA to make a DNA. Yeah, very bad at drawing with that little thing on my mouse. Okay, so reverse transcriptase produces DNA from the RNA of the genome. So HIV is an example. It's in the family lentiviruses that use this RNA or reverse transcriptase to convert its RNA genome into DNA. Now, reverse transcriptase is not an enzyme that we have on any of our cells. So when we were looking to fight retroviruses that use reverse transcriptase, one of the first attack targets was the reverse transcriptase. I mean, it's not something our host cell uses. Let's incorporate a drug that kills all the reverse transcriptases. And that's one of the targets for anti-HIV drugs is that specific reverse transcriptase. So once the reverse transcriptase changes into DNA, that DNA can then jump into the host chromosome, and we call that a provirus, and it can be replicated right along with our genome, just like it's a piece of our DNA. So that is the retroviridae lentiviruses like HIV, and then oncoviruses, which cause cancer, like HPV. Too much talking in the last couple of days. Okay, this is just giving that exact same thing I just talked about the retroviruses and the viral DNA, and then using the reverse transcriptase. It's just a nice picture summarizing that. Here's the reverse transcriptase there and how that gets incorporated as a provirus. So this is just a nice picture of what I just talked about, the retroviruses. Okay, maturation is now that we've assembled all the virus parts, genome, capsid, let's put them together into forming an intact virus. So creating those virus particles. And then once we've created a virus particle, the virus then needs to release to go infect other cells or to exit the host and infect another host. So in order for the virus life cycle to be complete, the virus needs to release, okay? Naked viruses, so viruses, again, that don't have an envelope, will release via cell lysis. So cell lysis means breaking open the cell, destroying the cell. Envelope viruses will release by budding or exocytosis because they need to take a portion of the membrane with it, right? The naked ones don't need that envelope, so they don't need to keep the cell membrane intact, where the ones that want to steal a portion of it need to keep it intact. Oops. Okay, this gives you just a picture of that budding process. So here is the viral proteins here, and they're attaching to the host cell membrane here. And here is the nucleocapsid. So that's genome plus capsid proteins. And the capsid is going to be surrounded by the host membrane as it's leaving the cell. And you notice here these little red guys, those are the viral spikes. And you can think of it as the virus particles and proteins are being made, one of the things that's being made is those viral spikes. And they actually insert themselves into the host membrane before the virus is going to leave. So those are not necessarily normal host proteins, but they're proteins that were created during the virus process and now are going to be part of the free virus particle. Okay, I'm going to post this video on Moodle so you guys can watch it. It's like I think it's from PBS. It's showing influenza. And the first part is really fun because you can watch a person sneeze and all the little particles come out of their nose and little viruses. It's kind of fun. Anyway, you guys can watch this as a good, it just shows kind of attachment and release. It's kind of the whole viral life cycle. And it's more kind of like a movie thing. It's not so textbooky. It's kind of fun. All right. So damage. Viruses infecting us, do they always cause damage to our cells? Do they always make us sick? Do they always cause some sort of disease? Most of the time, the virus will kill the cell or damage it so much upon entry that yes, it will cause enough damage to cause some sort of disease. Some cells can potentially become, stay infected and the virus doesn't release. And we call that period where the virus kind of hangs out, not really replicating, but is still there, hanging out in our cells. We call that latency. Okay? One example of that is the herpes viruses. Herpes zoster causes chickenpox, and you guys are all lucky enough to probably have had the chickenpox vaccine, so you didn't get to have chickenpox when you were a kid. I was unfortunately born way before you, so I got chickenpox when I was a little kid. And herpes zoster is a virus that caused chickenpox. And I clearly don't have chickenpox right now. I got it when I was 6 maybe, but there is still herpes zoster viruses living in my body. If at any point my immune system becomes compromised, severely compromised, it's potential that that virus will start replicating again. It has been latent in my cells. It hasn't been replicating, but it's just hanging out. And that disease that reoccurs from herpes zoster is called shingles. So with shingles, it's a potential that that virus can reemerge. Even the disease even looks different. Chickenpox versus shingles is very, very different. But that virus has been hanging out in my body for a long time in that latent state. And this usually reoccurs in older individuals because as we get older, your immune system starts to decline. Other viruses that we put into the classification oncoviruses can permanently change cells, and those are the ones that cause cancer. And normally how that works is that the genome will integrate into our cells, integrate into a portion of our genome where it controls cell division. Our cells will start dividing out of control, thus causing cancer. And HPV, human papillomavirus, is an example of that one. This is a nice graph showing the idea of latency. So if we look at this, this is the number of virions means how many viruses are in the body. So up here is going to be higher. The first stage of viral infection we call the acute infection. That's the first stage and sign of symptoms. So for chickenpox virus, this would have been, you get the little pox marks everywhere. It's really itchy. You get a high fever, and that will last for a couple of days, maybe weeks, depending on the type of virus. Then your immune system is able to combat that down, and the virus goes down to almost nothing, depending on the virus. But a few will lay low in your cells. And that latent infection might cause a little bit of increase in the virus, but not a lot, not enough to cause that acute symptoms again. And then again, if your immune system goes down, is compromised like with shingles, you'll have a resurgence of that virus causing a new type of infection in the case of herpes zoster. This is just a table of those different types of infections, talking about latency. HIV is one of the big ones that stays latent for years and years and years. You can get a primary infection that you don't really even notice, and then you won't start seeing the decrease of T cells for maybe 8 to 10 years after that initial infection on there. But there's just a couple of examples of the latent and then persistent infections. I'm just going to go over cancer really quick as we go into different. If somebody doesn't pick HPV, you can pick HPV. If somebody doesn't pick HPV, we'll go into cancer a little bit more detail. So oncoviruses are the ones, again, like I mentioned, that integrate themselves into the host genome, changing the replication of the cells, so that a cell that would be normal starts replicating out of control. HPV normally causing cervical cancer, it infects the cells of the cervix, potentially infecting a cell that hops into the genome in a gene that has to do with replication. The cell starts replicating over and over again, causing cancer. But HPV is not the only one. There's other ones that are able to do that same thing. The idea that it disrupts the host genome enough to cause the cell to replicate out of control. Also, retroviruses can fall into that oncovirus category as well. All right, so let's get to the star of the day. We're just going to talk a little bit about this virus. I'm happy to answer any questions that you guys might have. I will say that there's probably a lot of bad information out there, and there's a lot of information that is actually changing on a daily basis. And I'm going to give you guys, well, CDC, you guys know that website. I'm going to give you guys another website too that really has a lot of good information. Unfortunately, we're not going to know a lot about this virus until probably years after this initial kind of big surge dies down. But we're collecting information every day. It's changing every day, but we do know some things about the virus, and the scientific community is being really good about releasing this data and having a lot of this data available to everybody. So normally you would have to pay for some of these publications, but people want this information out there. They want to do their best to combat it as much as they can. Okay, so coronaviruses. Coronaviruses are a large group of viruses, and they're called coronaviruses because the spikes on the outside look like this pretty crown. So that's why we call them coronaviruses. They are enveloped. They're about 100 nanometers in diameter. They've got those spikes on the outside. We call them club-shaped. They are single-stranded RNA viruses, and the RNA itself, without the envelope, without anything, has been found to be infectious. Not that RNA is going to be laying around, but there's potential that the RNA itself can be infectious. The nucleocapsid is helical, and we think it is mostly infectious through vertebrates. I'll talk a little bit about that. We have seen transmission from the fecal oral route, meaning that there has been viruses in the feces for coronavirus, not necessarily the one we've been talking about. that can infect other individuals. Normally, coronaviruses are responsible for respiratory or gastrointestinal disorders, so mostly respiratory. I'm almost guaranteeing that everybody has had an infection with somebody from the coronavirus family. It is about 30% of the common colds are in this family of viruses of coronavirus. So most coronaviruses are just that, a common cold. So you can think everybody's had a common cold. Stuffy nose, coughing, you kind of feel like crap, may or may not have a little bit of a fever, but not a very severe fever. Couple of days, you're probably feeling A-okay, right? That's most of the coronaviruses. There are 3 so far, and this novel coronavirus that just came up is the 3rd of those that cause something more than the common cold. And we consider it a very severe respiratory infection. The first one of those, which was, I have it on my other slide, I think 2002 is when it first came out, was called severe acute respiratory syndrome, and the virus is called SARS-CoV. That's the first one here. So called severe because it caused a severe respiratory virus infection. The second one being the Middle Eastern Respiratory Syndrome, or MERS-CoV. And then this brand new one, SARS-CoV-2. And the disease itself is called COVID-19. That's, people use them interchangeably, but the official name for the virus itself is the SARS-CoV-2. So meaning that it is related to that original virus, the SARS-CoV, this one right here, and it shares in 79% similarity in the genomes. So we're considering them the same virus, just 2 different strains of that SARS virus, okay? Where the MERS was different enough that it got his own virus name there, okay? So let's talk a little bit about SARS. I'm planning to have a little bit more information for you guys too. If somebody wants to pick one of these viruses, you're more than welcome to pick them too. So this was in 2002, 2003, kind of similar to what we saw here. There was a couple of cases internationally. It started spreading. The cases for this virus were much lower than what we're seeing now. There's about 8,000. The death rate was pretty high, so that's what people took notice. It was between 9 and 11%, so about 700 of those people died. It did start a panic, kind of like we're here now, but it never got to the numbers where people were considering quarantining or shutting down anything, because it is far less than what we're having right now. The MERS was also a novel coronavirus. This one was different than SARS in that it was not being transmitted human to human, but from animal to human. I think it was called the camel flu when it first came out. I think that's what they were calling it. There was about 2,000 cases, so way far below what we're seeing now, but the death rate was super high. But as we're seeing with the death rates now, they think they're kind of inaccurate because there was probably limited people that are tested, people that had very, very mild symptoms never got tested for this. So when we're saying 2,000 cases, the actual number could have been 100,000, but we didn't know because at the time we didn't test that many people. And that's kind of what's going on right now. We are starting to learn more about what happened in China because it's been a couple of months. They were testing more people. You could see pictures on the news of people getting the drive-through testing. We're not doing that right here in the US right now. And they have just this week started amping up the testing. So we're going to start probably getting a little bit more cases as the tests are going through. And if you look at the CDC website, you can see how many cases are still pending on, was it human to human transmission? Where did the people get it? So there's still a lot of tests out there right now. So what do we know so far? I'm gonna give you a little bit of snapshot of what we know. So the disease is the COVID-19, the virus is the SARS-CoV-2. December 2019, so last December is when we first saw this pop up in China. We still don't know exactly how many cases, because again, not everybody was getting tested. But we do know that that is where it started. We started getting this high death rate. Now, a lot of times we'll see these viruses popping up in China and the Asian part of the world. Couple of reasons for that. They've got a lot of people in China, a lot of people, a lot more people than we do. And they tend to live with animals, not just dogs and cats, but like chickens and pigs, and live in the same space as those things, where we can get this change in host range, like with influenza, from swine to human or from birds to human, because that's where people are living in close contact. I don't know about you guys, but I don't sleep in the same house with the pig, but in parts of China or parts of Asia, that is a prevalent practice. And so that chance for the virus to go from one host to the other, from an animal host to a human host, there's a greater likelihood in places like that where you're living in close proximity with livestock. The current analysis shows that it's the SARS-CoV-2 is most closely related to a virus previously identified in bats. Now, as we do more analysis, if we find more viruses, that may change. We might find one that's more from a different, a virus from a different host. In the SARS case, it was a particular cat called a civet cat is where they found that the virus transferred from that host, the civet cat, into humans. So it was that leap from that host range into humans. As far as symptoms, so if anybody has any of these, immediately go to the doctor. No. As far as symptoms, so the biggest number one symptom with this virus so far we've been seeing is fever. So if you have a fever and it seems like the first couple of days, and as we track more and more people that have this disease, this may change a little bit. The first couple of days is kind of low-grade fever and then eventually spiking. The second symptom, number one symptom with most people is cough, and it's a dry cough. So normally when you have kind of like mucus, we consider that a wet cough, where a dry cough was not any sort of mucus in there. Some people will have runny nose. Some people will have diarrhea, vomiting. It really depends. But the two number one symptoms are the really, really high fever and the cough. Really quickly, if you did have one of those symptoms, one of the things we can rule out is that we can do a pretty rapid flu test. Again, flu is still out there. Influenza is still out there. Influenza didn't say, oh, COVID-19, you got this, I'm good. I'm gonna hang out and not infect anybody right now. So it's still out there. Respiratory syncytial virus, which causes severe respiratory symptoms, is still out there. It's still a virus that you can catch. It's still a virus that you can have. So there's a lot of other things that you can have that have almost similar symptoms. But it's a good, if you have flu or RSV or COVID-19, all of those things is good for you to stay home and not interact with anybody else. You don't want to pass those on to anybody. So the symptoms are similar, but it's good practice no matter what you would have to stay away from other people. Right now, the current testing for it is happening through samples. Most of them are being collected nasally, so they'll stick a swab way up in your nose, and they're going to test for the viral RNA. And they're going to use a process called reverse transcriptase polymerase chain reaction, or RT-PCR. Most of you guys have probably done PCR in either genetics or cell molecular right now. It's kind of the similar process, but instead of using Taq polymerase, you're using a reverse transcriptase like we find in retroviruses. And they're trying to determine if you have RNA for the virus in that nasal swab. It's coming back with a lot of false negatives sometimes. The PCR is not picking up everything. We're getting better at the testing, but it does take a couple of days for that RT-PCR to come back. And that's why the reports are not always exactly up to date. Not everybody is getting tested. Now they're getting more and more tests out there, and more people will be getting tested. So we might be seeing some spike in the numbers as more and more tests get out there. But that's the general idea for the test, is looking for the viral RNA. I didn't put any slides in here. We'll probably talk about this because I've already talked all right this long time. I can't believe I talked this whole time. We'll talk a little bit about why it infects older people and people that are immunocompromised. We can talk a little bit about that on Thursday when I talk about this a little bit more, and then influenza. So if you want to know right now, a really good website is from Johns Hopkins, and this is a slide that kind of I took this morning. I'm going to try to go to the website itself, and we can take a look at it if you guys are interested. If I can figure out how to share this. Let's see. Where's my little thing? Okay, let's see. Share computer sounds. No, I don't want to do that. Oh, wait. Let's stop share. Oh. Everybody's still there, hopefully. Share screen. There we go. Share. Yes, everybody can see this? Maps? Okay. So this has the total number of cases on it, in case you wanted to know. This is, again, from Johns Hopkins. So the total confirmed cases right now, they have 190,000 or so. And if you scroll down through these, you can see every city, every country and what their current numbers are. And it goes from most to least. And, you know, because China was the first place that it found, that's where the most cases still are. Italy is still pretty high up there. It has the total numbers of deaths and then the number recovered. So if you do the math on it right now, you can see it's pretty high death rate. That's going to change. It's going to change as we get more and more numbers out there, as we get more and more testing out there, as we find out more and more about what is, how many people are out there. So that number is going to change as we do more and more testing out there. But if you guys wanted to know some really accurate numbers, they are pretty good about getting the most accurate numbers. So you can see in California right now, we're at 11 deaths. You can zoom in on this little map right here, and you can get specific areas. Unfortunately, this bubble right here just is all California. It's not specific for our area. Again, the numbers right now are probably not very accurate because we are not testing enough people. Okay. So, why can't I see everybody? Everybody went away. I know you're here somewhere. Where are you? Oh, there you are. Okay. I'm going to unmute everybody. If anybody has a question, I'm happy to answer it right now. You can also type. I did see a couple of people typing answers. I don't know why they didn't pop up on my screen. But if anybody wants to ask. Anything. Let's see, where's the chat? Chat. Hi, everyone. Okay. I saw the chat. Anybody have any questions you want to put on the chat? I'm welcome to answer them. I'm going to post this once I figure out how to download it later today on Moodle. Again, no other assignments. I will do another video on Thursday so that you guys can hopefully zoom. All right. Any questions? Okay. Feel free to email me too if you have some questions. I can respond to you. We can do another individual one for everybody. Anybody who's got questions. Anything else? Stay safe. Don't go outside if you have fever. update you guys as soon as I can on the cloud situation. All right.
What?
All right. You guys on Thursday.
Bye.
Bye.
Adiós.